several key points. I'm going to give an overview of several key points in evolution, in our understanding of evolution in recent times. And so I'm going to talk a little bit about Darwin and Mendel. I'm also going to talk about work done on the structure of DNA. And then I'm going to, in the latter part of the talk, I'm going to focus more on genomics, epigenetics, and essentially the ENCODE project. So to give a brief intro, so of course in the mid-19th century, Charles Darwin published this bombshell book on the origin of the species and brought forward this argument that species evolve over time through natural selection. So this the theory goes that individuals within a population vary in different traits, and some of those traits are heritable. And those individuals that have the traits that best suit their environment survive and reproduce at higher rates. And so there's different individuals in a population. They have different traits, and the most successful ones pass the traits on to the next generation. And this is best shown in his analysis of the Galapagos finches. So Darwin, as a young man, managed to get on uh, a voyage of a ship called the Beagle um, that was going to go around the world. And it's very interesting because he was not very habituated to traveling overseas. He was sick many, many times when he started off on his journey. But he eventually got to the Galapagos Islands and observe these finches. And so what's so interesting about these finches? Well, the Galapagos Islands arose um, and became actual islands very recently, a few tens of thousands of years ago. And so these islands were taken over by different types of animals. And what Darwin observed was that the finches in different islands actually had different shaped beaks. And he thought, well, this is very interesting because maybe some of these beaks are better adapted to the habitat on each island. And so this is just a really interesting um, observation that sort of helped cement his ideas about evolution. And some people have actually gone and followed up on this. So you may be familiar with the book The Beak of the Finch, which was a Pulitzer Prize winning book from a few years ago. But in the late 20th century, Peter and Rosemary Grant followed up on these finches, did a long-term study on Darwin's finches in the Galapagos, and showed that actually, in times of scarcity, those finches which have beaks which are sort of best adapted to food obtention in a particular island, do actually tend to have a larger number of offspring, and that does have a significant effect on evolution within a few decades. And so it's absolutely fascinating that, the Darwin's finches, this was an initial observation, but it's been followed up, and there's substantial data to back up this idea that, that these finches are clearly a product of natural selection. We fast forward to slightly later on in the 19th century, Mendel at the same time was working on this problem of heredity. Um, Darwin's comments about natural selection are very general comments to do with, how evolution sort of is observed within populations over a large span of time, but it doesn't say anything about how the traits are passed on to the next generation. And so Mendel did careful experiments with pea plants around the same time that Darwin was publishing The Origin of the Species. He was doing these experiments. Um, it's it's uh, it's just absolutely amazing how detailed these experiments were, and he essentially showed that things are inherited independently. So, for example, whether a pea is wrinkled or smooth or yellow or green, these are sort of independently inherited in this sort of um, diagrammatic pattern that you can see here. And so what's clear is that there's this thing which we would now know as a gene, and that these genes are segregated independently and have a certain distribution in the offspring. And what's absolutely fascinating is that Darwin and Mendel were living around the same time. We tend to think of Mendel as slightly later because he was only rediscovered at the beginning of the 20th century, so a lot of people ignored Mendel's work. What's really interesting is Darwin actually recommends, at one point in his life, he recommends a book that was written by Mendel, but he apparently didn't read it. So he'd recommended this book by Mendel, but he apparently had not actually appreciated some of these things. It would have been very interesting to see how Darwin would have engaged with what we now know of Mendel's experiments and with what we now know about genetics. Um, Oh, this is absolutely fascinating. In Darwin's defense, I will say that peas are a very specific example to understand heredity. There are far more complex phenotypes to look at. There are far more complex traits to look at. And of course, in this wrinkly pea and smooth pea example looks very nice in high school textbooks, but actually, heredity out there is more complicated. Things like height, things like um, for example, hair color are actually a lot more complicated and they don't necessarily segregate very clearly. Or, for example, whether somebody has curly hair, that doesn't necessarily segregate particularly clearly in this kind of diagram. And so life is a lot more complicated. And Darwin was working mostly, well, he was interested in mammals, he was also interested in earthworms. And so none of those models fit particularly well with this, and aren't exactly peas. And so they don't fit really well with Mendel's presentation. And so later on, in the middle of the 20th century, a group of scientists, and this is a highly a partly tragic part of science history because there was work done mostly by Rosalind Franklin and Raymond Gosling, which was then provided by Maurice Wilkins to James Watson and Francis Crick. And essentially, the result of all of these interactions um, was that there were several papers published on the double helix and on the structure of the double helix. Of course, there's debates about, I mean, the real issue is that the Nobel Prize was eventually given for this discovery to Watson, Crick, and Wilkins, but Rosalind Franklin had passed away by then. It's a very tragic story because Rosalind Franklin and Raymond Gosling had actually done most of the work, the experimental work, and so this is a slightly fraught part of science history. The model that resulted from this revealed that DNA consists of two strands. So this particular two strands on the outside of the double helix, and on the inside is held together by interactions between the nitrogenous bases, so adenine, thymine, guanine, and cytosine. And so, and these match very specifically, so A with T and C with G. And so there's this major groove and the minor groove. So the double helix isn't perfectly symmetric, as you might see in subsculptures. There is some difference between the major and the minor groove. And it's held together by this sugar phosphate backbone that's on the outside. So one of the key take-home messages from the double helix is that when a cell reproduces, actually, there's one strand from the helix and the other strand will separate, and then there'll be a copy mechanism. So it's a little bit like double bookkeeping in the sense that you do have one strand and its complementary strand. So you can sometimes, I mean, it is easier to copy than certain other models that were proposed at the time. So DNA replication, the way the copying of DNA, for example, in heredity or for example in cell division, have a very clear model. And so this was a very early take-home message from the double helix. And so with all this information in hand, uh, we knew about the double helix in the mid-20th century, and towards the end of the 20th century, there were strong advances in molecular biology and DNA sequencing. And so scientists, as you know, can nowadays sequence entire genomes, thanks to mostly to work done in the Human Genome Project. And the Human Genome Project created a first draft of the human genome, so a first map of the human genome. And what's really interesting for the discussion today and from the perspective of evolution is that protein coding genes, so these genes that control things such as wrinkly or smooth peas, or for example, all the shape of the beak of a Galapagos finch, So protein coding genes are actually only 2% of the genome. So most of the human genome is actually not protein coding genes. And there's a large amount of non-protein coding regions, which actually were once dismissed as junk DNA. So it was thought that these regions were not important. This was mostly junk. Um, Now, later on, it was found that these regions actually um, contain regulatory elements. So, for example, promoters, enhancers, silencers, insulators. So, contain switches. So, they basically contain switches that switch genes on and off, that make genes active or inactive. And so it seems that a lot of the genome is mostly really important, really functional switches, and that actually the protein coding sequence, which is also important, is only 2%. So feel free to ask any questions if this um, if this is unclear. I mean, essentially, most of the human genome is a set of switching mechanisms that control a very small percentage of what are actual genes. And here's a diagram showing a sort of brief history of genetics. And as you can see, a lot of stuff, a lot of the Human Genome Project happened between 1990 and 2003. And so there's a lot of progress. The mouse genome was also analyzed around the same time. And so people started asking questions, when the human genome was completed. So we have a genome now, and there's a lot of hype, the people saying, when we have the human genome, we'll be able to understand human disease, we'll be able to understand human evolution. So when the human genome was first mapped, the people were saying, okay, so what's next? How do we understand? We have all these protein coding genes, we can understand this 2%. How do we understand the rest? How can we understand all of these switches? And so the government of the United States started this project called the Encyclopedia of DNA Elements. So trying to understand all of these different switches in DNA. And so this Encyclopedia of DNA Elements project, also known as the ENCODE project for short, essentially had the purpose of mapping all of this junk DNA, well, previously known as junk DNA, but essentially non-protein coding genome. And so essentially, this project had the focus of systematically mapping the biochemical activities across the genome. And what it showed is that a large part of our DNA is actually transcribed. There's a lot of chemistry going on in a lot of these regions, which are outside of these genes. And so a lot of these switches are active. They have proteins binding them. They have a lot of control mechanisms. So they're very important regions of the genome, um, despite the fact that they don't necessarily contain genes, but they do contain a lot of switches in them, which can be organized in a very complex manner, which can essentially result in very sophisticated gene control. Can you explain more about what mapping means here? So yes, I will explain this. So when we say mapping, um, when we were talking about mapping the human genome, that's when you get these large sequencing machines, and they essentially read all of the letters of the DNA using sophisticated chemistry. And so when a very similar process is used for what is termed as mapping junk DNA or sequencing junk DNA, so or non-coding regions. And so what is done is you can combine essentially sequencing. So the seq here, can you see dash seq? So there's a lot of technologies that you can combine sequencing with antibodies, for example, ChIP-seq, so chromatin immunoprecipitation. So you use antibodies and then you sequence them. And so these antibodies can pull down specific chemical modifications, which means that you can use the antibodies combined with the sequencing to essentially map what regions of the genome have a chemical modification. And so you can map the chemistry that's occurring across the entire human genome using a combination of two very simple elements, antibodies and DNA sequencing. And so fundamentally, yep, more questions. The regulatory DNA can affect morphology. So why is it not considered genes? That's a very good question. So, so genes in this particular presentation we're giving today, so we're being very specific that they have to encode a protein. Okay, so they're the parts of the genome that are read and essentially um, coded into proteins. However, there can be regions of the genome which essentially control whether those proteins are expressed or not, and those are switches. And you could say, well, those switches behave a little bit like genes because those switches, for example, if I delete one of those switches, I can end up with a completely different animal. And that is correct. However, in modern genetics, those are in I suppose if you're looking at the just at the animal at the end animal result and you're looking at whether it changes, you would maybe think that or name that as a gene. But in modern genetics, genes are only those regions of DNA that encode a protein, and regions of DNA that are switches, regulatory switches, are not called genes. Now, this is a very big issue because they're not genes, but they're obviously very important. So we need to map them and we need to be able to name them. And so there's an entire focus right now in genetics in terms of understanding and mapping and naming and characterizing um, regulatory elements, so switches in DNA. Um, and these are very important. And we have essentially names for different types of these regions, such as enhancers or locus control regions, and they're essentially being characterized. However, in traditional genetic terminology, we wouldn't consider them genes. They were considered, for example, regulatory elements. And so that's essentially a separate category, even though they're very important and they can affect how an animal, for example, behaves or how an animal, for example, how an animal looks like. Um, they are considered regulatory elements and they're considered separate from genes. And so so essentially all of these parts of the genome which are not protein coding but which are very important, they were mapped by the ENCODE project at the National Human Genome Research Institute. And to understand DNA, we need to understand that, those famous chromosomes, which are always shown on, when we're talking about DNA, these chromosomes, when a cell divides, well, these are highly compact strands of DNA that are wrapped around nucleosomes. So DNA is always comes in the eukaryotic cell in cells of mammals, for example, comes wrapped around histones, which are these proteins which essentially are used for packaging the DNA. And so the DNA double helix will be wrapped around histones, which are these proteins which are sort of packaging proteins, and these histones can be wrapped more or less tightly. And each sort of circular structure of these histones is considered a nucleosome when it's around by the DNA. And so DNA is in this sort of very densely packaged environment, wrapping around these proteins. And when it's read, so when DNA is active in a particular cell, DNA is less tightly wrapped. And so DNA is more accessible, right, to for example, proteins and to other elements of of the cell machinery. And so those accessible regions are known as hypersensitive sites. The regions which are essentially that can be bound by other proteins, they can be accessed. It's a little bit like a library. When a book is open and is actually being read, this would be considered in DNA terms as a hypersensitive site. And so there's a lot of chemistry that can be mapped around DNA, but the most important thing that you need to know is that ENCODE right now focused very strongly on hypersensitive sites. And these hypersensitive sites can be used to map all of these switches. So you can understand what switches are on and what switches are off in a particular cell. And so ENCODE used a lot of institutions, some institutions actually in Maryland. So as you know, Johns Hopkins was heavily involved. I was involved heavily in ENCODE in Seattle for a number of years. And um, this was a huge endeavor, right? Trying to understand all of these switches which are looping around in the cell and which are sort of activating genes and making sure that genes are actually translated or transcribed and then translated into proteins. And so this huge endeavor, right? Trying to map all these regulatory elements, trying to map all these things which are far away from genes, but then loop two genes in 3 dimensions and essentially switch them on and use very specific proteins to essentially achieve this task. And so once ENCODE completed, and ENCODE is almost complete, by the way. We are in the very final stage of ENCODE right now. Um, the key take-home message from all of this mapping and all of this regulatory element analysis across different species, different mammalian species, has shown that the key differences between species are actually mostly due to this junk DNA. So mostly species change um, in this intergenic DNA and outside of genes. So most changes between species occur outside of genes. They occur in these switches, and these switches that have huge alterations in evolution. So there's constant re-switching occurring throughout evolution. Um, and this makes sense from an evolutionary strategy perspective, because changes in genes can often result in damage, a big damage to the particular organism that has that change. There can be damage in survival, a gene can be switched off, and there can be huge problems in development. However, tinkering or changing this junk DNA, which turns out to actually be quite useful, tinkering in these regulatory elements in these switches is actually a lot safer. So it means that the animal in question doesn't necessarily have to um, develop aberrantly or die. Changes in these switches is actually, this tinkering of these switches is actually a much better way for evolution to achieve its aims. And so essentially, there's a huge amount of tinkering in terms of switches. So just to give a bit of a background, when we compare human and mouse maps for these gene switches, um, about about a third of the switches are completely repurposed. So that means that essentially a regulatory element or an intergenic switch that is used in the mouse, for example, in one tissue, so for example, in mouse liver, turns out that it's actually repurposed in the human and is used in a completely different cell type in the brain, and essentially still has a similar function in terms of its chemistry, in terms of its immediate chemistry, but has a very different function in terms of its implications for the particular organism which it's found in. And so this constant rewiring of genomic switches, and this is an extremely advantageous evolutionary strategy. So evolution in mammals is essentially the evolution of regulatory switches. So there's very few changes in genes. It's mostly changes in these control elements. And so essentially there's a lot of flexibility in evolution. So this is just a diagram from one of the ENCODE papers showing that when you have all of these DNase hypersensitive sites, so these hypersensitive sites I mentioned before, these switches which are open, around 40% of them are essentially um, have no sequence homology in human, but essentially about 35% do have both activity and sequence homology in human, and then some of them do not have any sequence homology. Sorry, do not have any activity in human, but do have sequence homology. And so, as you can see in this diagram here in panel C, you can see that some of these switches are the same when you compare human and mice, but some of them, for example, the orange ones are the same, but some of them are completely different. These blue ones are essentially completely different switches, which have essentially arrived there mostly because of the phenomenon of jumping DNA. This idea that this concept that there are transposons or parts of DNA that jump around and carry these switches. So one of the main reasons that people labeled the parts of the genome that aren't protein coding, one of the reasons that they're called junk DNA is because they have a lot of jumping elements. They have a lot of transposons within them. However, and so a lot of people are saying, well, those transposons, which are known, which originally came from viruses, are probably junk because, how is a virus going to contribute to evolution? Well, it turns out that those viruses are very useful for evolution because they actually carry a lot of these switches around. And so these sequences that used to be viruses are actually now parts of the human genome that move around and create a lot of flexibility for gene activation and for switches to be reused. And so fundamentally, the story's been flipped around. This idea that most of the human genome is junk, it's actually not junk. And it's actually really useful, and it actually repurposes mechanisms from retrotransposons to essentially, which used to be endogenous viruses, to essentially acquire a lot of regulation and fine-tuning of cell types, of cell type behavior, of essentially different properties within cells in the human body. So extremely useful stuff, and not at all junk DNA. So if there's one thing that we can take out of this discussion is that there is, yeah, the junk DNA is not junk at all. Um, so I'm going to move on to one of my projects when I was involved in the ENCODE consortium. I mean, I'm still involved in the ENCODE consortium, but this was one of the projects that I led within the consortium. Are there any questions first from the audience? Any more questions, comments? You can also speak in the chat if you want to ask any questions. So so fundamentally, I was involved personally in as I mentioned before. Oh, question. What is the practical use of this for mankind? That's a very good question. So so many of you, for example, will have heard of cases where there's a rare disease. So some families, there's a child, for example, that has a rare disease, and many times they cannot diagnose the gene where that child has that rare disease. So that child has a mutation, and we cannot find the gene uh, where that mutation occurs. And nowadays, if that happens, that's probably because it's not in a protein coding part of the genome. So mapping all of this regulatory DNA is not just a fascinating exercise of essentially the modern descendants of natural history. This is actually very important because we're actually mapping crucial switches, which are then important for actually clinical classification. So if you have a rare mutation in one of these genetic switches that is actually, uh, some of these switches have a larger effect, some of them have a weaker effect. But some of these switches, when deleted, can have a very strong effect. And so, if somebody has a mutation in one of these genetic switches, that could help understand why that particular individual has that disease, and potentially what downstream genes are affected by it, and what potential treatments can be applied. And so this does have very strong practical implications, not just in rare diseases, but especially in common diseases. So, for example, parts of the genome that increase risk of body weight, the parts of the genome that increase risk of a range of diseases, for example, schizophrenia, all kinds of other diseases. Most of them seem to occur in these switches. So most of those parts of the genome that affect your risk for disease are happening inside of the switches and are not happening inside of the genes. And so that's to understand those, we need to understand the switches in DNA. More questions? So are Mendel studied P genes, actual genes, but the Messier genes for, say, height or hair color are controlled by multiple genes or epigenetics or both? It's a very good question. The answer is both. So when you have very complex phenotypes like hair color or, for example, human height, which is incredibly complicated, those are controlled by essentially um, hundreds of thousands of different letters in the genome, some of which are spread across, most of which are spread across switches, and some of which are actually found within genes. And so I'm glad that you asked about human height. Um, so so those are very important advance in in genetics recently, which I don't think made it into the mainstream, but was very highly shared among scientists. And this was the mapping of genes that are involved in human height was actually completed in terms of saturated in 2022. So a study was done with around 5.4 million individuals, and we essentially now know how to predict all of your genetic predisposition to a particular height. And so height is partly genetic, as you know, and we now have a complete map of how you can predict human height in particular individuals. So they can take your DNA sequence and they can predict all of your genetic predisposition to a particular height. Height has been, in that way, solved genetically. So genetics of height is solved, and that's mostly happening in switches. Are most cancers due to mutations in regulatory elements? That's a very good question. So, some of the mutations that are predisposed to cancer, many of the mutations that are predisposed to cancer happen in these regulatory elements. That is correct. Not all of them, but some of them. There are mutations in cancer mostly occur in growth hormone pathways. So, for example, you may be familiar with BRAF and RAS, oncogenes. So there's different pathways that control growth in the cell. And so mutations in these genes are very commonly linked to cancer. However, there are also switches that control these genes, the expression of these genes, and the expression of other growth-associated genes. And so the answer is mutations can happen in these genes, but can also happen in the switches. And they tend to happen in both. Is it still believed that the human genome is roughly 6% of viral origin? I would actually put that number slightly higher. There's a huge amount of the human genome which is formed by retrotransposons. So retrotransposons and other types of transposons were famously discovered or suggested by Barbara McClintock, which was a very famous American, essentially geneticist. And she was doing studies mostly on maize. And what she showed is that there are parts that you will all have seen, or some of you may be familiar with different with maize where there's different coloring happening between the different essentially parts of essentially the corn husk. And so the difference in color that you're seeing there is because there's genes which are jumping, the idea that there's jumping genes that move within the DNA. And so what Barbara McClintock showed was that these retrotransposons exist. She was one of the first people, she was actually the first person to suggest that these jumping genes exist. And so these jumping genes are mostly originate from what used to be viruses. And so these jumping genes replicate around the genome and they tend to copy themselves many, many times. And so these jumping genes form a huge amount of human DNA. And sometimes you will read in the media, for example, that the pine tree genome is huge, right? So if you look at the pine tree genome, right, it's essentially 25.4 gigabases. So it's about 7 times bigger than the human genome. And so you'll say, okay, how can the Chinese pine tree, right, have such a huge genome, right? I mean, what is so complicated about the Chinese pine tree? And the answer is, it's not so much that the Chinese pine tree is that complicated, but rather it has lost essentially a fight between parts of the genome that are trying to avoid these retrotransposons or jumping genes jumping around too much. And the jumping genes have essentially gone crazy, and there's a huge amount of copying going on in essentially in these retrotransposons, and so you end up with an absolutely massive genome. And of course, this reaches constraints because I mean the genome can't be infinitely big, and so you have to copy all of this DNA. And in plants, especially, there isn't very much of a constraint compared to animals on the genome replicating beyond a certain size, and so you end up with huge genomes in certain plants. But that doesn't mean that, for example, the Chinese pine tree is therefore more complicated. It just means that. And it doesn't really mean that there's a lot of switches going on. A lot of the when a genome becomes that big, it's because there's a lot of these dead switches that are just copying themselves, and so there's a lot of inactive regions of DNA that are just extending themselves and copying themselves. So they're inactive in terms of function, but they do copy themselves at certain points in the life cycle, and so that essentially is why you have such massive genomes. in some of these plants, but it doesn't mean that therefore these plants are complicated or no. I mean, some of these plants are fairly simple in the biology. It's just that they've lost the war against the jumping viral genes, and so they're just growing beyond a reasonable size. Are there any relationships between particularly long-lived species and the genome? That's a very good question. So, if this question had been asked about 10 years ago, I would have said that we don't really know. But nowadays, actually, there are some recent reports um, on mutation, and specifically on somatic mutation rates. So this paper by Alex Kagan, based in the UK, he actually showed that if you look at different species and what their mutation rates look like, you can actually end up building a model um, where essentially the longevity of the species, and can you all see this figure here? It's a little bit grainy. Panel C, you can build a model where the lifespan of the species and its mutation rate are essentially form a curve. So for example, you have the mouse and the rat and the ferret, which have a very short lifespan, as you know, and also have a very high mutation rate. And then you have other species which have a very low mutation rate, for example, humans, and they have an extremely long lifespan. And so there are some, this is a very early report on this. This only came out a couple of years ago. It is thought that there is a relationship between mutation rates and species longevity. However, this is also controlled by epigenetics. You may have heard of the epigenetic clock, which is also a factor in this. So there's a lot of really complex chemical processes related to aging and why certain species age and why certain species don't age. Um, I was personally involved in a paper that we were doing on essentially the biggest effort to date on understanding epigenetic aging and different mammals. So you will find my name on this list. There's a large number of scientists. And essentially, what we did is we looked at 348 mammal species, and we mapped both the age of the samples and the chemical characteristics of the epigenome. And so what we found in this huge study that we did is that you can build one equation which essentially takes some chemical information from each mammal and can predict its exact age. So aging across mammals can be solved in one equation, one single equation. And so clearly the chemical marks and how we age are very can be mapped in a very simple way, looking at the epigenome. And so therefore, if we can use strategies to try to maintain a healthy epigenome, to try to slow aging or delay aging, then obviously that would be extremely beneficial. So we're really starting to understand a lot of the biology of aging, and I was happy to be a part of that story. What causes a high or low mutation rate? It's a very good question from Grace. So, so the answer to this is not entirely understood, and I'll explain a little bit why. So in this study that Alex Kagan did, they had to find a cell in the body which was a good gauge of mutation rates, because as you know, for example, if you take a skin cell in an average human, depending on the amount of sun exposure, there'll be a lot of mutations. So mutation can be affected by sun exposure, it can be affected by smoking, it can be affected by all kinds of behavioral aspects. So mutation can be affected by a lot of different factors. And so what they did in this particular study, and this is what's interesting, is they actually looked at intestinal crypts. So they took samples from the gut of different mammals. And there's some very specific cells with a very specific rate of division. And so they're sort of maybe a better gauge of the clock of mutation rates than, for example, taking a cell which has been exposed to sunlight, such as a skin cell. Um, so mutation rates are determined by environmental factors. So for example, as I mentioned, ultraviolet radiation, smoking, they're also affected by what the cell type is. So certain cells don't really divide very much, and therefore don't really mutate very much, such as neurons, and other cells are constantly dividing and are subject to slightly higher mutation rates. And so there's also all kinds of substances that can affect mutation rates. For example, um, you may have heard of aristologic acid, which is a plant which is found in fields. For example, it's very commonly found in the Balkans. And the reason that aristologic acid is very interesting in this particular regard is that if you have, for example, this was common in the Balkans back in the late 20th century. If you have bread and you don't have proper milling procedures and you don't have ways to separate the wheat from from the plants that have aristologic acid, then aristologic acid on its own will cause mutations, and there'll be specific cancers that are caused, and they can be mapped all the way back to this plant. And so there are certain substances out there which systematically cause DNA to mutate in a very specific way. And so essentially, what's important if we want to maintain mutation rates low is we have to make sure that we have a healthy diet, and that we're essentially careful with what we consume. This particular example of aristologic acid is relevant to the Balkans, but is also relevant to essentially parts of the world which practice traditional Chinese medicine. So certain weeds which are used in traditional Chinese medicine also increase mutation rates in a very specific way. And so you can actually sequence the DNA of the cancers that come from these people, and you can actually determine that it was originated by some of these plants. And so, I mean, I'm not saying that all of traditional Chinese medicine is going to be bad for you, but do avoid any plants which are higher in aristologic acid, because there's a focus on trying to detoxify some of these traditional Chinese medicines, but I would just say generally try to avoid any plants which have aristologic acid, since it does tend to mutate DNA in a very specific way. Yes. Any more questions or comments? Well, we've covered a lot of territory. I might go ahead and talk a little bit about my work in the ENCODE project. So I was focused very specifically on building a global index of mouse DNA and comparing it to human DNA. We're trying to understand how evolution happens. So there's at least 65 million years of evolution separating humans and mice. And so I was focused on the evolutionary aspect of this. We took a number of samples throughout mouse development. And so we had a huge number of samples covering different days of mouse development and fundamentally, and different tissues. And fundamentally, what we built was a map that goes all the way from mouse stem cells all the way through early development in mice, late development in mice, and adult mice. And so what we're trying to build here is a model to try to understand, for example, disease-associated regions, parts of the genome that are involved in diseases which are conserved between human and mice. And we're also trying to understand how brain development occurs. So, for example, there's certain genes such as NeuroD2, a very important neuronal gene, which are activated at certain points in development, for example, day 14.5 of mouse development. And essentially, we're trying to understand how mouse regulatory DNA works. So obviously, when we replicate samples, we have a very strong consistency. And what we found was very similar to what was previously reported in the first round of ENCODE, of the ENCODE project analysis. A lot of parts of the genome or switches are used across different organs. Some of the switches are specific to a particular system, for example, the respiratory system, or tissue, for example, the lung. And a certain chunk of switches are actually specific to a particular time point. And this is very interesting because some switches actually only become active in certain parts of development, and then they're never used again. So some DNA switches are used for essentially building a mouse and making sure that the mouse structure is correct. And then afterwards they're never used again. So there's a lot of regulatory DNA. It's very important in development. And it's also relevant for disease. And this is something I'm going to comment on slightly later in the talk. We showed that our maps covered a lot more DNA and a lot more switches than any previous study. And we characterize these switches across every single tissue. So we have forebrain, we have heart, we have liver. We characterize all the genes associated with each switch. And we built an atlas with 1.8 million regulatory elements to try to understand how the mouse genome works. Um, there's huge variation over time. So as you can see, there's a lot of active switches which go up later in development, and a lot of them are actually closed down. And transcription factors, which are these little genes that work around the genome and actually make sure that each part is doing it's playing its role. Transcription factors actually help understand how these switches work. They're orchestrators, or they're essentially a little bit like conductors in this orchestra of what it is the genome. And so when we looked at the brain DNA, we focused very specifically on neuronal samples. We could see this huge amount of change, right, from very early development to late development. And there's a huge shift in a specific point in development, which is essentially day 11.5 to 14.5, when the mouse goes from being sort of more embryonic type tissue to being more fetal type tissue. And so this huge change can essentially be mapped to the level of individual regulatory elements and binding sites, right? So these certain proteins bind to the genome and create a groove, and we can map all of that at a very high resolution to a specific base pair in the human genome. We can map all of this to an individual letter of DNA. So absolutely fascinating. And I'm going to go very swiftly throughout certain sections of this talk, but what I am going to say is that essentially we can use the regulatory maps that we have, the transcription factors, the maps from lung, the maps from heart, and we can use these to align human and mouse development. So we have all of the regulatory DNA that's active, for example, in mouse brain, and then we have data from human samples, for example, from miscarriages, and we can essentially map all of the human DNA, all of the mouse DNA, and essentially see how human and mouse DNA align completely from a regulatory perspective, which is absolutely fascinating. And we can see what programs are driving each stage, and we can try to understand diseases from this perspective. So, for example, there's certain mouse samples which are prior to any human sample that we have, and we can see that very specifically, for example, schizophrenia-associated variants, parts of the genome associated with schizophrenia are actually active in this very early stage of mouse development, for which we have no equivalent human sample. And so the whole question of whether we can use, for example, iPSCs to understand how those genes work and how those genes affect the context of schizophrenia, or whether we can use organoids. This is just really there's a lot of possibilities, and it's just absolutely fascinating that we can map these things nowadays. So yeah, so one more question. Can you speak to how epigenetics affects macroevolution, evolution over 100 million year timescales? That's a very good question. So so epigenetics affects macroevolution in the sense that these large programs that we see in mammals, for example, these large switches that we see are very characteristic, for example, of mammalian evolution. They create a toolbox for macroevolution to use. And so, for example, in bacteria, where none of these, so bacteria don't really have a large amount of switches. Bacteria are a lot more simple. They evolve mostly at the level of genes. There are some switches in bacteria, but it's not as common as in mammals. In bacteria, macroevolution will occur mostly on gene sequence and on very proximal, for example, promoter regulatory elements. Whereas in mammals, macroevolution occurs at the level of DNA switches at over very large timescales. It allows for very complex forms to arise. It allows for a much more complicated symphony to be played over the macro timescale, and that means that it's very different from what we see, for example, in bacteria. Um, these particular timescales, the toolbox is created there, a different toolbox. How is schizophrenia identified in mice? Or do I understand correctly that genes involved in schizophrenia in humans were identified in mice? So mice and humans have almost so a very large amount of human genes are conserved in mice. So if we talk about the genes, the protein coding genes, and these just to be clear, these were parts of the genome that were associated with schizophrenia in humans, and they were then conserved in mice. So these parts were identified in humans, and then we found out that they have the same DNA sequence in mice, and then we looked at how they were active. So we're not looking at the schizophrenia in mice themselves, we're looking at schizophrenia in humans, and then we're taking the DNA to its map in the mouse. Yeah, that was a good question to ask because it could maybe have not been clear if I hadn't clarified that. And how do you conduct comparative analysis of non-coding regulatory elements across multiple species? So you use DNA sequence. So DNA sequence can be aligned. You can compare the letters of the DNA and you can find letters of DNA that are shared. So if you have a part of the genome that is active, for example, in humans and active in mice and has the same DNA sequence or virtually the same, then you can talk about conservation and alignment. It's an alignment of the DNA. The figures on your slide are beautiful. Can you say what software you use to create figures? That's a good question. So I use Adobe Illustrator mostly, but there's also a lot of people are using different vectorial image software, such as, for example, Inkscape is a good option if you want something that is open access. I'm a big fan of open access, so yeah. And yeah, I wanted to say thank you very much, guys. We've done a long journey here from Darwin to the finches to all the way through the structure of DNA and large-scale genome mapping. So I wanted to say thank you to everybody. I think we've really tried to recapture a lot of the science in the last 200 years that has taken us to this point. And I really think this is appropriate for Darwin Day. I think Darwin would absolutely love this stuff, and I think he would be very, very keen on it. Yeah, and thank you very much, everybody. So if there's no more final questions, I can pass the um, microphone to Matt. Yeah, do you want to comment?
I have nothing to add to that. Thank you very much, Charles. Dr. Charles Breezer, Rouen University College London. We covered. You got that all in an hour, and that's fantastic. And it was all very clear and very much appreciated. So thank you so much.
Thank you very much, guys, and really appreciated the questions. And I think it's just absolutely amazing. Yeah, I want to say definitely take a chance to see the mammoth, and we'll definitely talk more about natural history. Yeah, thank you very much, everybody.
Thank you. All right, take care, everyone. The Fossil Club is here first Wednesday of every month, so join us again in
Thank
March.
you.